get us thinking about is this. I have a case study in plant speciations. So what about if we be able to use those concept in actual research and figure out if the plant species are different. And in this, I like to borrow the example from my friend. This is Dr. Tracy Misewicz. She's Polish. Well, no, she's American. Her last name is Polish. And in case you don't realize, she's super tall. She's my friend. We went to grad school together, and she's like 6'2". Like she's tall, and the whole family, like if you like, there's like three brothers and two sisters, and when they're together, it's like a wall. Like, hi Tracy, hi Tara, and like, and this is Tracy at work, and Tracy had the coolest field work ever. This is her PhD, and she climbed trees. So basically, she studied this plant called Proteum subseratum. The flowers. This species doesn't exist in Thailand. She worked in Amazonian Peru, so she have to fly to Peru half a year every year to study this plant, and the fruit look like this. And this is what the forest in Amazonia look like. There would be like typical forest soil, and this white sand patch next to each other, like they're like mixing. There's two types of soil, and this species of plant, they look kind of different. There's like this morph, the white sand morph, like the white sand morphology, and the brown sand morphology. They look kind of different, but not very different. So her advisor, Paul, who's also interesting person on different occasion. So Paul discovered these two morphs in Peru in his PhD, and Tracy now working with Paul. And Paul wanted to see if they become two different species yet. So Tracy had to go to Peru, and she basically on the watch, like she on the email, and her assistant would be like, hey Tracy, it's flowering right now, come down right now. Like she had to fly from California to Peru, like, and she like have 2 days, and she climbed up the tree and back the flowers, and to see if she prevent these two species from mixing with each other, what would happen. Like, and she had to like hike like 9 or 10 kilometers each day to get to the trees and climb up that tree. And she installed the camera up top of the trees to see who's visiting her flowers, like, and she had to identify those insects by pictures. So it's a lot of work, and Tracy spent like 4 years working on this. It's like one of the craziest PhD I ever seen. Like, Tracy, you're crazy. And she's like, yeah, I know. And yeah, I mean, it happened that her boyfriend then and husband now, who else, he happened to be in my lab, so I get to know a lot about Tracy's story. And Tracy was like, if he proposed to me in Amazon in Peru, I gonna drown him in Amazonia River. Like, I'm gonna kill him, because like this is like the least happiest time of my life. So those of you who like romanticize working in the forest, having like in touch with nature, Tracy would tell you differently. It's like, this is the worst time in my life, basically. Just like anybody who tell you having babies best thing in their life, they're lying. Like, babies would take all your times. So okay, that's Tracy. So what I'm doing right now is that, so I'm gonna give you some figures from Tracy's dissertation. This is the actual figures from her dissertation, and I want you to figure out what kind of isolation, or is it pre-psychotic or post-psychotic, or is it what type of isolation? And I take out the mechanical, because like plant mechanics always work with each other. So I'll have 4 figures like this. Figure one is you have 2 types. You have parental cross, which is basically same soil, have the same soil, like brown soil to brown soil, white sand with white sand. The hybrid is the white sand made with the brown sand, and they adhere pollen grains to the pollination, how much of the pollen grain actually attached to the flower. And number 2 is the flowering times up to populations. And figure 3 is number of seed per pollination. between parental cross and hybrid cross. And finally, table 1, look at the pollinators and the white sand. So what I'm gonna do here is to have you work on your own, not in groups, because I know you're sick of breakout rooms from other classes and this class. So I'm gonna give you about 10 minutes to look at this, and also as a break for yourself, so you can walk away a little bit and think about this 4 graph, and I'll share the poll, the second poll. You have figure 1 to 3 and table 1. So take about next 10 minutes as a break, and also think about this, and when you're ready, you can submit the poll and we'll come back and go through the answers together. All right, so it's 3:10 now, so we'll be back at 3:20. Okay, so think about those, or step away for bathroom break and whatnot. So we'll back at 3:20. So I'm gonna end the poll now. I think most people are already in. There's some variation in the answer, which is good. All right, let's see. Let's share the results. Can you look at the result as well? Okay, let's look at the first one here. So the first one here, you see that the adhere pollen grain for pollination, basically, so what Tracy does is do the hand pollination. So this is a female part, right? And I'll use the purple one for female. And then the pollen here can come from either the same soil or different soil. It's a parental cross or hybrid cross. And if it adhere, meaning it start making pollen tubes. So this is almost like egg and sperm meeting with each other, right? And they start recognizing each other. It's the same species. And you can see that the hybrid cross, meaning like you get the pollen from different population, tend to be less successful. And the p-value here is your statistical test, like t-test, is less than 0.5, meaning it's significantly different. So the hybrid cost is a lot less, not a lot, but like significantly less successful than the parental cross, meaning that this preventing them from mating with each other. But this is the example of, most of you put it correctly, this is gametic isolation. Right, and some people say ecological, it could be ecological, but in this case, we forced the two cells, like two male and female sex cells, to meet with each other. So this is a case of gametic isolation. They not yet formed the zygote. The next one is the flowering times, and you can see that both white sand and brown sand are actually flowering at the same time. So this is Tracy trying to test the temporal isolation, whether or not the flowers come out at the same time. But what happened is that they actually flower at the same time. So this is what Tracy tried to test, but it turned out that they're not really happening, right? So the temporal isolation did not happen in the system. So she showed that like they flower at the same time, at the peak at the same time. So it's no time prevention in this case, but that's the test for temporal isolation. And this one is the proportion of seed. So in case your plant biology is not the strongest game, the seed is the product of fertilization, right? The fertilization had to happen first to produce a seed. So seed is the embryo of the plant. So this happened when the zygote already formed. So she wants to see if let's say the pollen do adhere and germinate and fertilize with an egg, would it yield the viable offspring? And basically, the production of seed in plant is equal to a viable offspring. And you can see here that the hybrid cross, when the brown sand made with white sand, tend to produce less viable offspring. So this is a case of a hybrid breakdown, actually, which only 3 of you chose. And the reason is not the hybrid infertility, is because the hybrid infertility would be that you get the seed, right? And you use that seed to germinate again, and that seed will become infertile. So that would be hybrid infertility. This is a case of hybrid breakdown. It's not even form a seed. So you can see here in the hybrid, only 15% produce the seed, as opposed to the parental cross that more than 40% produce seed. So this is when the example from real life really different from the example from textbook, right? Because in reality, it's not black and white. It's not either you produce viable offspring or not. It's more like if you are the same species, you have higher chance of not failing. You can think of this as human too, like human we mate all the time, right? But some of us having miscarriage, some of us having a baby inside, but baby die before even born. So not all humans are successful at reproducing. So same thing here, that even they are the same species, same population, it does not always produce viable offspring, but they have better chance that to produce the viable offspring than the individual from different populations. So this is a case of hybrid breakdown. And finally, the table. So the table show that the visitor for flowers, if you brown sand, you most likely get the B, morph C. But if you are the white sand tree, you get mostly morph A. And you'll be like, why don't she identify them to the species level? Like, why can't she tell the species name? Like, she get this data from the video, so the bees sipping in for 2 seconds, and like you have to be able to identify them for 2 seconds. So like, yeah, the morph A and morph B is the best you can do, really. And you ever try to identify bees? They're pain in the butt. Like, it's super difficult, and to like all different length of the links, and oh, not fun at all. So, but in the end, like, you know that C is different from A, and you know that if you in brown sand, you get a lot more different types of bees coming visit your flowers compared to the white sand. So this kind of separate your pollinators, and that's testing the existence of ecological isolation. So this is a real example of how people like Tracy, who is like evolutionary biologist, she used the idea from biological species concept to test whether or not these two becoming 2 different species already. And she, in her thesis, she concluded that these 2 populations is about to become 2 different species. So like, she caught them in the act, basically like, you guys are splitting, I know it, because like there's a couple of different isolation already happening, and she's detecting right where it happens. This is a rare case of like you be able to spot, like you caught them in action. This is like not very often you get to be at the moment of things speciating, and this is one of them. So, but as we seen before, like not all biologists, especially people like me, who like more on the systematic side of things, we tend not to like biological species concept too much. They're like, what, why? But it's so good. Like it's so clear and it's so mechanistically sensible, right? You can test them, you can like see if the species are different or not. But the problem with species concept are a few, and I'm going to give you a few examples here. So the first problem is like it relies on, sorry, it should be plural, rely on sexual reproduction. So the whole biological species concept rely on reproductive isolation, meaning you want the organism to have sex. If if the sex is not happening, then you don't get this. You don't get to define the species concept, right? And the second part is that the problem with hybridization. Like somebody said earlier, what if you have chimera, like the two organisms actually made and make viable offspring that look like half, like you made the, I don't know, the rhinos and the horse together become unicorn. What if unicorn exists? What you gonna do, right? Like what you gonna name them is so now the rhinos and the horse become the same species. So like what you gonna name them? Like the whole biological species concept is kind of broken apart. And the sexual reproductions is the key here, is that some species don't do sex at all. They like reproduce asexually. And imagine like the practicality of this. Like what if you want to be like you're systematic, right? And you want to like test whether or not the two giraffes are the same species. What do? You go to Africa, right? And you make two giraffes, like make one of them produce sperm, and you extract the egg of the other giraffe, and you make fertilize, and you wait 10 months for the giraffe to be born. Like really, that's how you have to do it? So like have to like make giraffe have sex, and you watch them like, yeah, do it, give me baby, give me some baby, you'll be alive. Like really, like so like if you have to do that every time you want to define species, you won't be done with anything. Like it will take you forever, right? And the hybridization, not so much in animal, because animals, when they cross different species, they tend to fail permissibly. But if you are plants, this happens all the freaking time. Like the hybrid plants are like one of the major industry in plant world. Basically, things like this. People name all these different species, but you found out later that, oh, look at the chromosome number, it's actually 24, which is half and half from this, and this species is half and half of this. And if you use, and this species, this hybrid are viable, they be able to reproduce and make their own offspring that look exactly like them. So in the end, it's sort of like, if you use biological species concept, all of this, dubious, miscellaneous, pretentious, myras, porifolius would be just one species, right? Because they can mate each other and produce viable offspring. Then the whole thing breaks down. And you go talk to botanists and they'll be like, but this pink and this is yellow. You tell me it's different, it's the same species. You crazy biologist, you crazy evolutionary biologist. Like that's not happening for me. Yeah. So in the end, it's like, what? Really? Like, and how do this? And then things like this, like orchids are a great example. Like, if you are at all in horticulture, people breed plant all the time to get things they want. Like cymbidium, this is Cymbidium nocative, or the tailor bird, is a heat tolerant cymbidium, because cymbidium, most of the orchid like the cool, moist environment, but most of the time the houseplant in Bangkok or metropolitan areas tend to be hot, so they breed them to be more heat tolerant. And Cymbidium nocative is come from these two different species, Dianum and Floridahasin. And Floridahasin come from another two parents that look totally different, and they kind of breed for the length of the petals and the pattern on these petals. So the breeders are really creative with this, and they breed them over and over and over and over until like basically it make no sense for mostly for botanists to be like, I can't use biological species concept because things just so readily to mate with another. Like they can't even do cross genus, cross genera. So biological concept for a lot of people just doesn't work. It's just like they reproduce with each other all the time. So I could present you the third and newer concept called phylogenetic species concept, in short PSC. So basically use that same concept that we learned before about monophyletics. So basically population of each species should share a common ancestor, or they should be monophyletic. Basically, the species is monophyletic group. So bypass all that, we know that morphology sometimes can be deceiving because things can look morphologically really similar, but they're different species, or morphologically really different, like dogs, but the same species. But like, let's not worry about that. And let's also not worry about sex. So you don't try to make two giraffes together because it's too crazy. It's too much work, and I don't have time in my life to watch giraffe have sex or not have sex. So let's use phylogeny to define the species. So basically, the species is monophyletic group, and be done, right? You guys know how to do phylogeny already, and you know how to define monophyletic group already. So just get the DNA and figure this out. So, and that's the reason why we know these three picture of these giraffes are different species. So they do morphology and molecular phylogeny based on DNA of giraffe, and found that the grouping based on the coat colors and also based on geography. So this cluster in this area used to be called the same species, but now thanks to molecular biology, they now be able to separate into different species here based on coat color and also by their populations, and the DNA confirmed their existence. So now we know the giraffe is more than, it used to be like a fewer species, but now they found the subpopulations that genetically distinct and also match with the coat color that people previously ignoring. So that's what we found in giraffe. And come back to the bear examples, I kind of teasing you before that it's actually maybe one or two species. And this is what happened. So remember last week when we do the practical, I asked you to do the chloroplast DNA and nuclear DNA and to see if we get the same result. And for some of you, it might not give the same result. And the red panda and the panda bears might probably jumping back and forth. And that's exactly what happened in the bears. So on the left here, we have the nuclear DNA, which put the polar bear and the brown bear as distinct group, monophyletic for both group. But if you look at mitochondrial DNA, it turns out that polar bear is still monophyletic, but now brown bear, have to circle the brown bear, I'll use different colors. The brown bear now is paraphyletic because it includes everything but this one clade of polar bear. So like, what? Like nuclear say one thing, mitochondria say one thing. Which one should I believe? Like, which one should I like? Should I believe you? Should I not believe you? Like, is mitochondria better than nuclear? Is it not? Like, which one is the right answer? There are different species. It turns out you have to think about the inheritance of nuclear DNA and mitochondrial DNA, right? Because mitochondrial DNA only pass on to the mother, but this pass on both sides, both mom and dad. So the fact that polar bears have mitochondria that looks like brown bear probably has something to do with the mother. So what they believe happened is this, that about 600,000 years ago, polar bear and brown bear start to diverge to become different species. And then some naughty brown bear during late Pleistocene, this is Ice Age basically. So when Ice Age happened, you know, the ice sheet formed between polar area and the not so polar area. So some crazy brown bear, some like adventurous brown bear, probably the mother, like some single lady brown bear, it's like, oh, the single lady, oh, the single lady, just like, ooh, it's cold out here. I'm feeling adventurous. Let me travel up now to see some white fur male bears up there and see what's going on and like check me out. And then like this naughty brown bear go mate and leave some mitochondrial trace within the polar bear. So the babies are still polar bear, but they have some mitochondria from the mother brown bear down south. So, and then since then, the ice retreat, and then they become they separate population. So there's some brown bear mitochondria left in the polar bear descendants. So that's what that seemed to be what's happening here. So can that kind of break down a little bit? So if you use biological species concept, the brown bear and polar bear are no longer distinct species because their ancestor at some point being naughty and start mating with each other. So no good. But phylogenetically, it's like, well, if you look at the bigger picture, like the nucleus DNA, they're still separating. And so that's why it's phylogenetic species concept tend to be able to teach this apart as well. So go beyond just mating problems. And it's especially useful for things like this, lichens. So this is the work group I work with during my postdoc. This is the species called Graphescripta. And it used to be just one name. And over the years, since it's being named, it's split into over 100 species, hundreds. And this is a really common species they found in Europe. And everyone who found new things, like, oh, they found a straight one, oh, this one species. They found a forky one, another species. Or not so forky one, another species. So they like keep giving new names. And in 1929, people were like, you know what, you guys are crazy. They just squiggly the things, or just one name, done, 1929. That's good, right? It's Graphescripta. Graphis means graph, just writing. Script means script, so writing. So like squiggly thing on the tree. So they just put it on one tree, one name. And then in 2011, there's some two crazy Dutch people. No, one crazy Dutch people. I probably should not say it out, not crazy. There's some inventive Dutch and German people who are my lovely colleagues. Hopefully they don't see this YouTube video. They probably won't. Don't send this to them. They know they're crazy. But yeah, well, they're like, you know what? We think there's 4 species in this, not 100, but 4. So they come up with morphological species concept. They were like, okay, if you are branching a little bit, then use species B. If you straight line, use species S. If you straight line but curve, you are P. So they start naming this thing. And my advisor, who like, I don't believe them. I think they're crazy. They're just making things up. These 2 crazy Dutch people are the crazy. Like they don't know what they're doing. So let's prove them wrong. Like, woo, that's exciting. That's science, drama and science. Let's prove people wrong. So like, he's like, I don't think their species is correct. And so I don't know anything about their rivalries or if they're not arch enemy, they're just disagreeing. So I do the molecular phylogeny, and it turns out that, so I asked all the data. I actually asked the specimen from those 2 Dutch guys. It's like, send me some specimen, I'll do DNA for you. And because you can wait for lichen to mate, like lichen, they have sex or don't, or they grow like 1 millimeter a year. So even if they have sex, you have to wait for 35 years for them to produce the first thallus. So like, we're not gonna do that. We're not gonna wait 35 years to see the result. So I have like a year and a half on my contract. So I have to figure this out now. So we found that there's like 5 groups of these so-called species. So what used to call Graphescripta is actually 5 groups, and not to our surprise, this is morphotype. Remember those like MSPB that I talk about, those 4 species that the Dutch guy talk about are completely a mess. So yeah, like 4 versus 5 be close enough, but his 4, like their 4 is completely messed up. So for example, my species 1 here correspond with their 3 species. So things that look like this could be the same genetic group. So morphologically, they don't look similar, but they are in the same genetic groups. So the morphological concept fail here is like, doesn't reflect the evolution. And this is where people start arguing, because like, but like I'll show you next picture. So and this is politics of science. So the way we deal with this, so we invite those 2 guys to work with us. So we sent him the result and like, what do you think? And he's got so offended. It's like, no way, I can, like, this is not right. You did wrong and like blah blah blah. And he finally gave in. So I put the grouping and the specimen together. I took all the photos, and like I basically look at them for months and like, what do you guys have in common in this group 3? And I basically like, are you guys the same species, or like you guys genetically similar but you look really different? What's going on? Is the morphology works at all? Because I have to be able to defend my result. Like, I think this is good result. Just that the morphology that you see that does not reflect the evolution. Because this is where people are going crazy. Because if you believe in evolution, in phylogeny, and you try to group them based on monophyly, then you have no morphological data to support it. And how are you going to tell what species is this? And they're like, psh. So like my job would be like, what put them together? Like, what do they have in common, if not the look? Because the look is not at all similar. So that black thing you see, the squiggly line thing, this is thing called ascoma. Is the fungi term. Is a sexual reproduction part of the lichens. So I look at ascoma morphology and I measure everything. So I measure the width and the length and the margin width. So the margin is this. Basically, they have a little margin here on the ascoma, and I measure the width of that margin. So I spend an awful amount of time. Actually, not my time. I actually have an undergraduate student working with me, and the poor thing had to measure everything. So that's the idea for your project, right? If you want to spend most of the time in front of your computer, you can do that. So you measure everything, and there's no difference, like no difference between 5 groups. Like, oh my God, yeah, like, yeah, division of labor, or more like just labor, their labor, not mine. So I'm like, okay, and the student left me, and they're like, and he quite deflated because like he was hoping to see the differences, and like, oh, and like he's like, I'm done, like I can't look at this anymore. Not France, like France did not do this, like France did another thing. Yeah, France doing something a lot more exciting than this. So this is actually back in my Chicago day. So they left, like he left, and they're like, okay, what else going on? So I cut open the ascoma and the spores. Like, hallelujah! The length of the spore, the difference. Group 1 and group 2 is longer spore, and the other, like, finally, thank God. Like, but still, like, I still can't separate one from 2 and 3, 4, and 5 from each other. So in the end, like, what did I do? Like, what's going on? So in the end, what really happened is that, so I decide at the end of the paper that I would put them back into one species, that those 4 morpho species that they put before doesn't make any sense. Like, it doesn't really correspond with phylogeny. But I did not want to split them into 5 species because we don't have the identifiable morphological characters to work with. So it's still, we're gonna come back to this result after the midterm, and this is a case of like what I call cryptic species. So species are there, they are different species, but they're kind of cryptic. They're not really clearly separating them apart, but genetically they look different. But we're gonna come back and talk about how we know that they're clearly different and why it matters. But for now, is a utility of phylogenetic concept that allow us to actually think about the species in the group that sex is not possible, or observation of the mating is not possible, or things that have really complicated sexual history, or like reproduction history. And that's example of the use of phylogenetic concepts. So that's, like, again, a tip of the iceberg. And like to say, like, this class is probably doesn't feel like systematic classes, feel like molecular evolution, biochemistry, and stuff like that. But that's the nature of modern systematics. It's a more integration of knowledge as opposed to memorization of what the groups are. If you ask all the systematist people, today they be like, well, the expertise in the group is important, but understand the principle behind the grouping is would allow you to do different groups, and you gain those experts from plant biology, animal biology, and microbiology, and probably hopefully things from this class will come add to that, right? So that's all I have for the lecture today. So